Good evening. Welcome to the last session of this course. We will wait for a couple of minutes before everybody joins in and then we will start. Stop transcription. Okay, good evening. Welcome to the 12th session of the 12-week course, Enzyme Sciences and Technology, which is taken by Professor Vishal Trivedi of IIT Guwahati. I'm Sanket, a research scholar from Kusuma School of Biological Sciences, IIT Delhi. So today we will be talking about enzyme applications, and it will be a continuation to what we had discussed in the last week, right? So as we already have talked about a few things, that enzymes can be used in case of agriculture, in pisciculture, that means for fishes. It can be used for poultry, vaccine development, for transgenic animals, medicine, genetically modified organisms, and for drug delivery. We already discussed enzymes which are required for the food industries, for let's say in dairy industry, or in making a clarification of juices, in wine and beer industries. Uh, Also in the medical field, we have discussed about how enzymes can be used as diagnostic markers. We can check the levels of enzymes like SGPT or SGOT, for let's say liver. They are markers for liver injury and things like that. Enzymes can be used for genetic engineering as well in a laboratory setup. We have various enzymes like the restriction enzymes, right? Restriction enzymes which can be used to make genetically modified organisms. We have other enzymes as well, like ligases. We have polynucleotide kinases and multiple enzymes which are involved. In terms of environment, there is like bioremediation. Uh, in case of bioremediation and other environmental factors, in such cases, enzymes are also essential. We'll talk about it. Now, in disease diagnostics, we can use enzymes, right? For example, glucose oxidase and glucose dehydrogenase can be used in case of detecting the blood sugar level, blood glucose level. in case of diseases like diabetes mellitus. So similarly, we can use glutamate, check the amount of glutamate decarboxylase for neurodegenerative diseases. And what it essentially detects is the neurotransmitter GABA, gamma aminobutyric acid. Similarly, enzymes can also be used for treating diseases. There is enzyme replacement therapy, which is we will be discussing about. It can be used for detoxification as well, for drug metabolism, for hormonal regulations, anti-inflammatory agents, and as biosensors as well. Now, talking about cancer, which is a diseased condition in a lot of cells. Uh, this table gives you a list of characteristics and what is different between a normal cell and a cancer cell. For example, in terms of cell growth, normal cells have a controlled division, whereas cancerous cells, they have uncontrolled division. In case of DNA repair, there are mechanisms which are in place and they work to repair damaged DNA. Whereas in case of cancerous cells, there are usually mutations in the gene which regulate this DNA repair pathway, which leads to genetic instability and there are more errors. Similarly, for cell death, we know that normal cells, they go for programmed cell death, which is also known as apoptosis, whereas cancerous cells, they can evade apoptosis, and hence the cells which were destined to die, they actually survive, and that creates a problem. Now, in case of telomeres, telomeres are the ends of a chromosome. The ends of the chromosome are the telomeres. Now, in the concept of aging, it is thought that telomeres, they shorten with each replicative cycle. And there is a limited amount of telomere which is present in our DNA or in a chromosome. So once this limit is reached, the cell can no longer divide and it has to go for apoptosis or senescence. Now, uh, telomerase is an enzyme which replenishes these telomeres. So this telomerase enzyme is not found to be very active in normal cells. However, in cancerous cells and also in stem cells, there is high level of telomerase activity that can potentially, in theory, let the cells divide indefinitely. That is a problem because when cells divide, they accumulate mutations and those mutations are carried forward. So that is a problem. Again, in case of cancer, in case of gene mutations, the mutations in genes that control the cell death or division is rare in case of normal cells. However, um, these mutations are present in especially in the genes which are called tumor suppressor genes or the oncogenes, which promote uncontrolled cell division. Now, talking about the causes of cancer, there are various causes. One is the environmental factors, which involves exposure to carcinogens like tobacco, UV, asbestos, arsenic, formaldehyde, can cause cancer to form. A poor diet and lack of physical activity can also contribute to development of cancer. Chronic inflammation and oxidative stress is a problem. Therefore, antioxidants is important. Inflammation needs to be reduced if it is like that, because in a lot of cases it is a problem. It can cause scarring of tissues leading to cancer. Now, there are genetic factors as well. There are mutations in oncogenes or tumor suppressor genes. There could be spontaneous mutation in the DNA replication, or there could be epigenetic changes, like in case of methylation, DNA methylation, which can contribute uh, to expression, dysregulated expression of genes. This can lead to cancer. right? DNA damage is another reason where there could be single stranded breaks or double stranded breaks. So single stranded breaks are usually caused by UV radiation or reactive oxygen species due to high oxidative environment of the cell, whereas double stranded breaks can be caused due to X-rays or gamma rays, which have high penetrating power. They can cause DSBs. Now, replication errors which happen due to multiple replicative cycles, if they are not rectified, can lead to DNA damage, which sustains in the cell. Now, uncontrolled growth, cell growth and proliferation is another reason, one of the hallmarks of cancer, which happens due to dysregulation of cell cycle checkpoints. So what happens? So we know that cell cycles, there is a cell cycle which is maintained by every cell, which has different phases. Like we have the G1, S, G2, then we have the M phase, and then it is a cycle basically. And these cycles have checkpoints before each of these before they proceed to each of these phases. These checkpoints are essential to ensure that the cell has adequate number of the cell is good enough or healthy enough to proceed to cell division. However, when this gets dysregulated, what happens? The cell usually goes for cell death or apoptosis. But if the cell cycle checkpoints are dysregulated, what happens is the cells which were destined to die, they actually survive, and that is a problem. They are insensitive to apoptosis. Apoptotic signals are there, but yet the cell are not receptive of it. Um, there could be increased apoptosis in healthy cells that could happen. So it will increase the population of the cancerous cell, and there is telomere shortening. So in case of infections, they usually cause inflammation. For example, in case of hepatitis B or hepatitis C viruses, they cause inflammation in the liver. Which leads to liver cancer. Similarly, there are viruses which integrate into the host cell, like there is HPV infection, which causes cervical cancer. There is HIV, which also gets integrated and can lead to multiple problems. Uh, Chronic stimulation by viruses like HTLV could be a problem, and bacterial toxins can also lead to cancer. Then there are oncogenes and tumor suppressor genes. So usually the gain of function mutations in oncogenes, example CRAS, HER2, MYC, BRAF, EGFR, and ALK, they lead to cancer. Similarly, loss of function mutation in tumor suppressor genes like PP53, P53, BRCA1 or 2, or retinoblastoma gene, they lead to cancer as well. Now, for cancer, there are several drugs which have different classes, as you can see in the table. So, like there are alkylating agents. There are alkylating agents, um, which like cyclophosphamide, cisplatin, and carmustine, which cross-link DNA to prevent replication and induce apoptosis. The target enzyme here is DNA polymerase, which actually adds nucleotides to DNA strands during replication. Similarly, there are anti-metabolites like flyfluorouracil, methotrexate, and gemcitabine. They disrupt DNA synthesis and repair by mimicking the naturally occurring nucleotides. The target enzymes here are thymidylate synthase, dihydrofolate reductase, and DNA polymerase. And they essentially are important for synthesis of nucleotides, which are needed for DNA replication. Similarly, there are topoisomerase inhibitors like irinotecan, etoposide, and doxorubicin. They inhibit the topoisomerase activity, topoisomerase 1 and 2 in specific. And these topoisomerase are is usually helpful in uncoiling of the supercoiled DNA strands. Similarly, there are mitotic inhibitors like paclitaxel, vincristine, and docetaxel. They disrupt the microtubules that prevent proper chromosome segregation. during cell division. And the target enzyme here is tubulin, and it forms microtubules required for proper cell division. Similarly, we have DNMT inhibitors. They specifically target DNMTs, which is DNA methyl transferases. Decitabine, azacitidine, and guanidinoacetabine, these are some drugs. They inhibit DNA methyl transferase activity. These are the enzymes which are used to methylate DNA. Now, talking about metabolic disorders, metabolism is the process by which the body converts food to energy. Now, metabolic disorder can occur when there is a problem in this process, leading to inability of the protein, inability to process certain nutrients or produce energy. Different metabolic disorders like diabetes, phenylketonuria, and galactosemia are known. There are many others as well. Symptoms of metabolic disorders can vary depending on specific conditions, but may include fatigue, weakness, weight loss, weight gain, and changes in appetite. Treatment may include dietary changes, medication, and or enzyme replacement therapy. And this is something which we will be talking about in detail. Um, Metabolic disorders can be diagnosed through blood tests, genetic testing, and diagnostic procedures. Now, talking about the disorders and their specific enzymes. For example, in case of phenylketonuria, the defective enzyme is phenylalanine hydroxylase, which converts phenylalanine to tyrosine. Uh, but a defective enzyme will not be able to convert phenylalanine to tyrosine, leading to accumulation of phenylalanine. Now, the therapy which is recommended is low phenylalanine diet, BH4 supplementation, Kuvan is a drug, and gene therapy. Similarly, in case of galactosemia, the defective enzyme is galactose 1-phosphate uridyl transferase. It converts galactose 1-phosphate to glucose 1-phosphate, right? And for therapy, there is a galactose-free diet, monitoring blood glucose, calcium, and vitamin D supplements. Then there is maple syrup urine disease, which the defective enzyme in this case is branch chain alpha keto acid dehydrogenase complex. It converts branched amino acids to its corresponding keto acids. What is recommended is low protein diets, thiamine supplementation, and liver transplant. There is Pompe disease, which uh, is caused due to a defective acid alpha glucosidase. It breaks glycogen to glucose, right? In this case, enzyme replacement therapy is helpful. Gene therapy and supportive care is also there. In Fabry disease, alpha galactosidase A is defective. It converts glycosphingolipids to globotriacyl ceramide. Here also enzyme therapy is used, can be used. Chaperone therapy or gene therapy can also be used. In Tay-Sachs disease, hexosaminidase A is defective. It breaks down GM2 ganglioside. In this case, supportive care, symptom management, and gene therapy is sought after. Now, talking about AIDS. AIDS is acquired immunodeficiency syndrome. It is a fatal condition which is caused by HIV, that is human immunodeficiency virus. It damages the immune system and leaves the body vulnerable to life-threatening infections like and also cancer. So virus enters the target cell, that is the immune cell, by binding to the CD4 receptors and co-receptors CCR5 or CXCR4. After entering the cell, HIV converts its RNA genome into DNA using the enzyme reverse transcriptase. Viral DNA is integrated into host genome by the enzyme integrase. Once integrated, the viral DNA is transcribed and translated into viral proteins, including the viral protease. This viral protease cleaves the polyprotein into mature viral proteins, which are assembled to new virus particles and released from the cell. There are antiretroviral drugs which target these enzymes, which are involved in the HIV replication cycle. They include reverse transcriptase, integrase, and protease. We will study about these drugs. By inhibiting the activity of these enzymes, antiretroviral drugs can prevent the virus from replicating and reduce the viral load in the body. While there is no cure for HIV or AIDS, retroviral drugs can be used to help people living with the virus maintain a healthy immune system and prevent the progression of the disease. For example, for the target enzyme reverse transcriptase, which converts viral RNA to DNA during replication, there could be nucleoside reverse transcriptase inhibitors, which are like zidovudine, lamivudine, and emtricitabine. These are nucleoside analogs which can actually prevent the reverse transcriptase to work. There could be non-nucleoside reverse transcriptase inhibitors as well, like efavirenz, nevirapine, and delaverdine. They can structurally impair the enzyme reverse transcriptase, not making it functional. Similarly, proteases which cleave viral polyproteins into mature proteins during the virus life cycle can be inhibited using protease inhibitors like saquinavir, ritonavir, and atazanavir. Integrase, which integrates viral DNA into the host cell, can be inhibited by using integrase inhibitors like raltegravir, elvitegravir, and dolutegravir. GP41, which is glycoprotein, which facilitates the fusion of viral and host cell membranes, can be inhibited using fusion inhibitors like maraviroc. Now, diabetes is another condition which we'll be discussing about. Diabetes is a metabolic disorder which is characterized by high levels of blood sugar because the body is unable to produce insulin or it cannot utilize insulin, is not receptive to insulin. Now, insulin is the hormone that is secreted by pancreas that regulate the blood sugar level. So diabetes can be of two types. Type 1 diabetes, where enough sufficient insulin is not produced by the beta cells because they are lost or damaged. However, in type 2 diabetes, insulin is produced, but the cells are not receptive to insulin. Right? These are the two types of diabetes. Now, high blood sugar level can damage the blood vessels and the organs, leading to a range of complications, including heart disease, stroke, kidney disease, and nerve damage and blindness. Enzymes, they play a very crucial role in the metabolism of glucose and regulation of blood glucose or blood sugar level. The enzymes are like alpha glucosidase, which converts carbohydrates to glucose in the small intestine. Similarly, there is enzyme like DPP4, that is dipeptidyl peptidase. It inactivates incretin hormones like GLP1 and GIP. These are incretin hormones, and when they suppress this hormone, it increases the glucagon secretion. It suppresses the insulin and increases glucagon secretion. Similarly, we have sodium glucose cotransporter 2, SGLT2, which, as you can see here, SGLT2 will help reabsorption of glucose from the kidneys, right? It increases the glucose level in the blood, or rather maintains it. Insulin degrading enzymes are there. Insulin degrading enzymes, they degrade insulin in the liver and other tissues, right? And similarly, these enzymes can be targeted, as you can see in this table, like alpha glucosidase can be targeted using acarbose and miglitol, which inhibits alpha glucose activity. DPP4 can be inhibited using sitagliptin and saxagliptin or linagliptin or leptin enzymes, drugs. They inhibit DPP4, increases GLP4 and GIP, and promotes insulin secretion. Sodium glucose cotransporter can also be inhibited using canagliflozin, dapagliflozin drugs. So talking about liver cirrhosis, it is a chronic liver disease which is caused due to progressive replacement of healthy liver tissue with scar tissues, leading to liver failure. So one of the Major reasons behind this is due to hepatitis B or C infection, which causes chronic inflammation or damage to the liver. There could be alcohol abuse, which causes cirrhosis, very common. And one of the most common ones, which is coming up now, is non-alcoholic fatty acid disease, fatty liver disease. Um, Very common in India and is an active research topic. Enzymes, they play a very crucial role in the development and progression of liver cirrhosis by breaking down the extracellular matrix, promoting collagen cross-linking, or metabolizing toxins and drugs, and generating ROS. So some of the enzymes that are important are like matrix metalloproteinases. They break down the extracellular matrix and allows tissue regeneration, remodeling, and repair. So similarly, along with MMPs, there is TIMPs, which is tissue inhibition of metalloproteinases. They actually inhibit MMP activity. Collagen cross-linking enzymes like LOX and LOX-like, that is lysyl oxidase and lysyl oxidase-like enzymes are also there. Apart from that, glutamate cysteine ligase and cytochrome P450 enzymes are also there, which maintains the antioxidant environment. So the drugs for MMPs is doxycycline. TIMP is up. TIMP1 is upregulated using NAC, that is N-acetylcysteine. We have already studied about it earlier. N-acetylcysteine also maintains a redox environment, a reducing environment, or antioxidant. It's an antioxidant, and it is also used to treat malaria in certain cases as a drug for malaria. Cross-linking enzymes like LOX or LOX-like, they can be inhibited by beta amino propionitrile. Right? BAPL. Similarly, glutamate cysteine ligase, which is an important enzyme for glutathione synthesis, can be again controlled or regulated using NAC, N-acetylcysteine, which increases the glutathione levels. Cytochrome P450 enzyme. They are responsible for metabolizing the drugs and toxins. Uh, they can be inhibited using pentoxyphylline. Uh, Alcohol dehydrogenase and aldehyde dehydrogenase can be this is used to actually process alcohol in the liver. They can be inhibited by disulfiram. Also, given this drug is given to treat alcohol disorders, people who have drinking issues, it's used to treat that. For neurodegenerative diseases, certain enzymes like acetylcholine esterase can be inhibited using donepezil, uh, galantamine, some these kind of drugs, which essentially it inhibits this acetylcholine esterase, increasing the acetylcholine levels in the brain. Similarly, monoamine oxidase can be inhibited by rasagiline, which performs a selective and irreversible inhibition of monoamine oxidase, reducing the breakdown of dopamine. Very effective in case of Parkinson's disease. Similarly, tyrosine hydroxylase can be inhibited, can be using levodopa, which is a precursor of dopamine. It crosses the blood-brain barrier and increases the dopamine levels in blood, again used for Parkinson's disease. Catechol O-methyltransferase. It breaks down levodopa and dopamine. It reduces the efficacy of these drugs. So the inhibitors would be entacapone and tolcapone. Which inhibits COMT. GABA aminotransferase is used in metabolism of GABA. Deficiency of GABA T leads to increased GABA levels in epilepsy. The drug is vigabatrin, which inhibits GABA T, increasing the GABA levels, gamma aminobutyric acid levels in the blood. Now, in case of environment, the enzymes can be used in paper and pulp industry and for bioremediation, wastewater management, and biofuel production. So enzymes like cellulases can be used, which hydrolyze the glycosidic bonds which are present in cellulose, lignocellulose, and hemicellulose. Cellulose is a crystalline structure of linear beta-1,4-D glucose. Hemicellulose includes xylins, xyloglucans, arabinoxylins, and mannans. These are essentially cell wall components in case of plant cells. Cellulolytic organisms like T-RAC produce three major enzymes: endoglucanases, exoglucanases, and beta glucosidase or cellobiase. So endoglucanases cleaves the internal beta-1,4 linkage, glycosidic bonds. Exoglucanases they hydrolyze cellobiose from the ends of polysaccharide chain, and beta glucosidase or cellobiase they cleave cellobiose and other oligosaccharides into glucose. Now the source is it's widely distributed because cellulose, a lot of plants have cellulose, hence cellulases are also widely present. The industrial cellulase which is produced is from a filamentous fungi, which is found in soil, plants, and marine environments. Many studies have shown various fungus to have cellulases in them. Similarly, we have chitinase, which is the second most common or abundant compound. I'm talking about chitin, not chitinase. Second most abundant compound, which is just after cellulose, and it is formed by monomers, by polymers of NAG, that is N-acetyl um, glucosamine. Polymers of NAG will actually form chitin. So this NAG polymers will be broken down using chitinase to oligo-N-acetyl oligonads, and then chitobiase breaks it down further to the NAG monomers. The commercial source is shrimp, um, crabs, and lobster waste for chitinase. Naturally, chitin is bound with protein, lipid, pigments, and minerals like calcium carbonate. Therefore, chitin preparation involves demineralization and deproteination of chitinous waste using strong acids and bases. Right. So if we talk about the application, they have huge applications in pharmaceutical industries. Pharmaceutical industry, they are biocontrol agents as well, and they are better than other biocontrol agents. So chitin is essentially it affects the chitin, which is a protective layer in case of insects. So it works as an insecticide, and it is better because it does not have any polluting effect. Um, in case of nutrition and parasitism of bacteria, because these are those bacteria which actually thrive upon these insects, they have chitinase in them. And they have a morphogenic function in fungi, protozoa, and invertebrates. They are associated with defense mechanism in plants and vertebrates. They can also control mosquitoes and for degradation of degradation of fish waste. Similarly, lipases, these are actually triacylglycerol acyl hydrolase. Right? They belong to the hydrolase family and they act on carboxylic ester bonds. Their physiological role is to hydrolyze triglycerides into diglycerides and monoglycerides. The end product is fatty acids and glycerol. Right? The reactions which are catalyzed by lipases are like esterification, acidolysis, transesterification, alcoholysis, and aminolysis. Right? They don't require any cofactor to function and possess good regioselectivity, chemioselectivity, and enantioselectivity. They exhibit a broad substrate specificity. Right? And they can work at various temperatures based on, of course, sources from where they can be isolated. So the source can be animal, plants, and microorganism. Lipases are found everywhere. The commercial lipase are actually sourced from yeast or fungus, and even microorganisms. One of the most studied form of lipases is pancreatic lipase, which is found in pancreas of obviously in humans and pigs. They are most studied and most documented. Uh, okay. Pseudomonas species also they also have lipase, which has versatile reactivity and stability in non-aqueous environment. So they work function in non-aqueous environment, and hence have a lot of interest. So talking about pectinases, there are 7 different enzymatic activities which contribute to breakdown of pectin. So pectinases basically break down pectin. They're obtained from natural ingredients to which is used to produce coffee and chocolate. Pectinases are also used in removing mucilage, which is developed during fermentation, right? Pectinases are also used, you have already studied about them for juice clarification. It enhances the antioxidant effects of juices, and it gives the color also, increases the colors. The most common pectinase is Aspergillus species. I mean, the source is Aspergillus species. It's a fungus, and that's the most common source. They produce pectinolytic enzyme, deacidifying and chain splitting enzyme, also obtained from tomatoes and oranges, although in low quantities. Now we will be discussing about the questions for this week. So the enzyme involved in the production of dopamine is GABA aminotransferase, acetylcholine esterase, monoamine oxidase, or tyrosine hydroxylase. So the answer to this would be tyrosine hydroxylase. The enzyme that converts viral RNA to DNA during viral replication cycle is topoisomerase. Tubulin reverse transcriptase or DNA polymerase. It's actually reverse transcriptase. Match the disorder with their respective defective enzymes like Fabry disease. Um, Fabry disease actually causes alpha galactosidase A, their defective enzyme. Gaucher disease by glucocerebrosidase, Tay-Sachs disease by hexosaminidase, and Pompe disease by acid alpha glucosidase. We've already discussed this. So A1, B2, C3, and T4. The role of catalase in food industry is to break down fats, to improve the dough texture, to reduce browning in fruits, or to decompose hydrogen peroxide. So catalase actually decomposes hydrogen peroxide into water and oxygen. So that's the function of catalase. So it is used to catalyze hydrogen, decompose hydrogen peroxide. The enzyme that is commonly used in the production of bread to improve the dough elasticity is lipase, amylase, protease, or glucose oxidase. It's actually amylase. Match the drug with their enzyme inhibited. So 5-fluorouracil will inhibit thymidylate synthetase. Acyclovir will actually inhibit DNA polymerase. Lovastatin will inhibit HMG-CoA reductase, and 6-mercaptopurine will uh, will be inhibited by adenyl succinate synthetase. So that's the code. So A1, B3, C2, and D4. The primary function of endoglucanase in cellulose degradation is breaking cellulose to cellobiose, hydrolyzing internal cellulose bond, converting cellobiose into glucose, or enhancing cellulose crystallinity. It's actually to hydrolyze internal cellulose bonds, in specific the beta-1,4 glycosidic linkage. Match the drugs with their mechanism of action. So vincristine is to disrupt the microtubules. Irinotecan is for to inhibit the activity of topoisomerase. Cisplatin for cross-link DNA to prevent replication and induce apoptosis, and then decitabine to inhibit DNA methyltransferase. So it's actually A4, B3, C2, D1. The organophosphates, which is commonly used as an irreversible acetylcholine esterase inhibitor in insecticides, is atropine, uh, pralidoxine, malathion, or parathion. So it's actually parathion. The end product of chitinase activity on chitin molecules is glucose, fructose, N-acetyl glucosamine or galactose. So we already know that chitin is actually polymer of N-acetyl glucosamine. So N-acetyl glucosamine would be the final product. The type of chitinase acts on internal beta-1,4 glycosidic linkage of chitin chain. We've already discussed about it. It's actually endochitinase. The end product of triglyceride hydrolysis by lipases is monosaccharides, amino acids, fatty acid, and glycerol, or nucleotides is actually fatty acids and glycerol. In pharmaceutical industry, lipases may be employed for antibiotic production, digestive system health, drug delivery, or respiratory support. So lipases can be used for drug delivery system because it can, penetrate the liposome barrier, the phospholipid barrier, which is there in the membranes. The effect of pectinase treatment on fruit texture is increased firmness, enhanced crispness, softening, or enhanced strength. It actually causes softening. The enzyme, which is often elevated in case of blood or in blood of individuals with liver cirrhosis, indicating liver damage, is creatinine kinase, alanine aminotransferase, amylase, or lipase. So it would be alanine aminotransferase, which always gets elevated whenever there is liver damage. So with that, we are at the end of this course. We have completed 12 weeks of this course. If there are any questions, you can ask them now. Okay, if there are no questions, then we can call it a session today. And I wish you all the best with the assignments and the exam that you will be giving for certification. All the best for that. Good luck. Thank you very much. With your permission, I will end the meeting now.